Sources of oligonucleotide primers include designing primers using softwares, retrieving pre-designed primers from published research articles, from various vendors, and from various repositories like websites. Today, we will talk about one such website called Primer Bank. Head to the website as shown in the screen below. The link to this website is also provided in the description below. This website allows users to retrieve PCR primers available in this repository. To search a PCR primer, select the appropriate option that would be used to retrieve the gene name of the primer. For instance, users have the option to choose an accession number, IDs, gene symbol, or a keyword. The easiest option would be to use the keyword for the gene name. In the species option, select either human or mouse accordingly. Since keyword was selected for the search mode, therefore, enter the name of the gene, for instance, collagen 6, and then click Submit. Clicking on the Submit button redirects to another web page. This web page displays the information pertaining to the primers. It returns the gene ID, accession number, name of the species, coding DNA length, and the gene description. The output displays various primer pairs of the same gene with different primer properties like amplicon size, primer length, melting temperature, and location of the primers. Many primer pairs are available, and users can select any of these primers according to the experimental conditions. Let us analyze primer pair 2 in detail that has an amplicon size of 166 base pairs. Click on the cDNA and amplicon sequence option in primer pair 2. Here, users can find the location of the primers and the sequence that would be amplified by the given primer. For instance, the red DNA sequence here is the site of the forward primer, and the one in the end is the site of the reverse primer. The nucleotides highlighted in blue is the sequence that would be amplified by the primer pair. Primer Bank website also offers users to select any specific splice variant or a transcript variant gene primers. For instance, this particular primer pair can amplify the collagen 6A5 transcript variant. Transcript variant collagen 6A2 is also available if users would like to select. Similarly, another transcript variant available is the collagen 6A1 mRNA. Primer Bank also offers validation of the primers. For instance, clicking on the validation results redirect to another web page that shows both semi-quantitative and quantitative real-time PCR validation of the primer pairs. At the end, this website has a feedback form. Users have the option to report whether the primer pairs work in their experiments. Thanks for watching. Please subscribe for more videos.